Hello, everybody, and welcome back to BioSC 140 Human Physiology. This is the part 2 video for group problems 2 for lecture exam 2. So we kind of stopped off in the very middle of this question because my power went out. Well, we're going to pick up right where we were. So we talked about how the resting membrane potential is created. We talked about how you can create EPSPs and IPSPs by changing the membrane permeability to different ions, by opening up sodium channels, opening up potassium channels. The question asks about the equilibrium potential for individual ions. If the membrane was permeable to only one ion, the value of the membrane potential would be equal to the equilibrium potential for that ion. It's like, what's the contribution of that ion to the overall membrane potential? That is, the equilibrium potential is the value when the electrostatic force exactly opposes the concentration force. So remember, we have electrochemical gradient. At what point for this specific ion does the chemical gradient equally oppose the electrical gradient? So that there is no net change in movement of the number of ions on one side of the membrane to the other. This occurs when the cell is permeable to one and only one ion. So the equilibrium potential for potassium is -90. The equilibrium potential for sodium is +60. So next up in this question, it asks about the action potential and what's happening with the ions during the action potential. So during the action potential, let's see, well, first I want to point something out. I do want you to know these numbers, key values you should know, nudge, nudge, wink, wink. This is an important section to study these right here. And so we just talked about the equilibrium potentials. We're going to get to these bottom 4 numbers next. All right, so down here, the generalized action potentials. We just talked about how resting membrane potential is set up. We talked about how you can have graded potentials, IPSPs and EPSPs, if you change the membrane permeability to different ions. If those IPSPs and EPSPs come together to create a situation where the cell depolarizes to -55 at the beginning of the axon, aka the trigger zone, aka the axon hillock, we're going to get an all or nothing event called the action potential. And the action potential happens because of voltage gated sodium ion channels on the axon. So when the voltage hits -55 at the beginning of the axon, those voltage gated ion channels, so the gate is controlled by the voltage of the cell. When that first gate opens up, it's going to start the cascade event that sends the action potential down the axon. So it's the -55 at the axon hillock. These voltage-gated sodium channels are going to open up, increasing the permeability of sodium to that portion of the cell, and sodium is going to rush in. We're going to add a whole bunch of positive sodium ions in a very quick, short period of time. And the membrane potential is going to shoot from -55 up to +30. And right about when it gets to +30, there's actually a second gate on those voltage gated sodium channels. So these are double gated channels. Double gated channels, sodium channels found on the axons. The first gate opens up, the activation gate opens up due to the voltage of the area, -55. The inactivation gate will close, closing the channel around +30. At that same time, we're going to have right up here at the peak we're going to have a whole bunch of potassium ion channels open up also. And so potassium is going to rush out of the cell. You're removing positive ions from the cell. It's going to drop the membrane potential actually all the way down below resting membrane potential before they close. And then we'll come back to the equilibrium point. Down here below resting membrane potential is hyperpolarization. Up here at the top is the peak. Threshold is where those first sodium voltage gated channels open. So you know those values, you're able to tell the story of the action potential and how ions are moving throughout this process right here. Double gated sodium channels, voltage gated sodium channels open, sodium rush in, inactivation gates closed, potassium gates open, potassium rushes out, they overshoot resting membrane potential before they close back up and come back. Between the differences between action potential and creative potential, this summary chart right here, definitely pay attention. Definitely pay attention to this summary chart right now. Let's just do the summary chart. Let's just do a brief overview of what this is. The action potential that we just talked about. This is process right here that's activated on the neuron that happens in the voltage gated sodium channel, propagates an electrical signal on the neuron. Those ions have to diffuse away from that channel. So with distance. Action potential is axon only. Graded potentials, you can actually find them on dendrite cell bodies, and you can find them in some situations on the axon. Coding, action potential, you can't change the amplitude, so it's frequency based. Faster, higher frequency or lower frequency of signal. Graded potential has the way it kind of codes information is the amplitude of the change. The amplitude of the change in potential. Channels, so action potential is double gated sodium. Graded potential is single gated sodium, calcium, potassium, or chloride ions. Refractory period, action potential has a refractory period. That's a period where another action potential cannot be sent. A period of time where another action potential cannot be sent. Graded potentials have no refractory period. Summation, there is no summation in action potentials. With graded potentials, you can summate. There can be many different ion channels opening up at the same time, which can cause summation. Active potentials are always depolarizing. Graded potentials are depolarizing or hyperpolarizing. Now open up a sodium channel, sodium rushes in, depolarizing. Open a potassium channel, potassium rushes out, hyperpolarizing. So please be able to, you should be able to explain the differences between active potentials and graded potentials for the exam. For the hypothetical cell questions, I'm not going to work through this step by step because I feel like this explanation really takes you point by point. And also in the lecture page for the nervous system, we have a number of different problems that really take you step by step through it. Um, so definitely look those over. Definitely just take it step by step. Step one, determine the resting membrane potential. How is the membrane potential going to change when the ions move the way they're going to move? Are you adding positives to the cell? Are you adding negatives to the cell? Are you removing positives from the cell or removing negatives from the cell? Determine the peak action potential. Take your time, follow your equation, do the math. All right, so draw a schematic of three major efferent fibers from the somatic autonomic nervous system include both sympathetic and parasympathetic fibers, including a diagram the relative length of each fiber, the non-ganglionic, preganglionic, and postganglionic fibers. Also labeling the diagram, making sure to include the location of cell bodies, name the location of any associated ganglion, the neurotransmitter released from each synapse, and possible types of receptors on the postsynapse. So this equation is pretty much the last video in the nervous system series of videos. And yes, it takes a good amount of memorization. I think the best way to learn this material, to practice this material, is by drawing out the summary chart that we have at the end of this page and at the end of the video, the normal videos for the nervous system. This right here. So this right here tells you everything that the question is talking about. Somatic or voluntary motor. Where are the cells found in the central nervous system, the cerebral cortex, ventral gray horn? There is no ganglion. It's one fiber in the somatic nervous system to the skeletal muscle. When you get to that skeletal muscle, the neurotransmitter is acetylcholine. The receptors are cholinergic, subtype nicotinic, part of the skeletal muscles. In the sympathetic nervous system, we have ganglionic and non-ganglionic. Ganglionic is the typical, when we think of the sympathetic nervous system, the typical one. So central nervous system cell bodies are going to be found in the lateral great horn and thoracolumbar. So thoracic spine, lumbar spine, they're going to have short preganglionic fibers. So the sympathetic nervous system is going to follow the two fiber rule. Short preganglionic to the ganglion, where the ganglion is a group of peripheral nervous cell bodies. Neurotransmitter. I remember this because I just so clearly remember seeing the sympathetic chain ganglia in anatomy lab that I can't forget it. I always remember sympathetic. The ganglion are really close to the central nervous system, short preganglionic. Neurotransmitter acetylcholine, subtype cholinergic, receptor cholinergic, subtype nicotinic. And then we have long postganglionic fibers in the sympathetic nervous system. We're going to have the neurotransmitter norepinephrine then, receptor type androgenic, subtype alpha 2s. We also have receptor androgenic, subtype alpha 1s, beta 1s, and beta 2s. So one thing I want to point out, these postganglionic cells, when they release their norepinephrine on type of cardiac muscle, smooth muscular glands, there's three subtypes of receptor on the target cell. And there is one subtype of receptor that's back on the postganglionic cell, the alpha 2s. So this cell releases norepinephrine, and that norepinephrine can bind back onto that receptor on the same cell. Alpha 2s prevent the over release of norepinephrine. They cause feedback inhibition. The non-ganglionic sympathetic is going to be going to our chromatin cells in our adrenal glands. So single fiber going to chromatin cells. Chromatin cells are actually kind of analogous to the postganglionic nerve cell. Instead of releasing norepinephrine onto the target muscle or gland, they just release norepinephrine, epinephrine into the bloodstream, so neurohormone. With the parasympathetic system, we're going to be craniosacral for our central nervous system cell bodies, long preganglionic fiber, neurotransmitter acetylcholine, receptor cholinergic, subtype nicotinic. This is going to be then our ganglion, and our ganglion is going to have a short postganglionic fiber going to cardiac muscle, smooth muscle, and glands. Transmitter is acetylcholine, receptor type cholinergic, subtype muscarinic. So make sure you practice drawing out this diagram right here. If you're able to do it all from memory, that will help you on the exam. All right, everybody, those are the 8 group problems. If you have any questions, please email me. Remember, I am here to help, and I will see you in the next video. Take care.